package one, this is all about understanding how tropical root knot nematodes are going to be potentially invading Europe as climate change takes hold and soil temperatures warm up. So we've been developing a new soil sampling and identification protocol to understand exactly which nematodes are where in Europe and enable researchers across the program to be able to enter their data into a database that we're going to be using then to make our projections as to what's going to happen in the future. We're approaching this from two sides of Europe, so we're starting in the west in Spain and moving up through France towards Germany, and we're starting in the east in Turkey and moving up through Slovenia and Serbia up toward the more northern countries like Germany and the Netherlands. Yeah, so we've made some really great progress in being able to identify these different nematode species using molecular methods, and that's a brand new and exciting result. We've also gathered together a huge database of observations of nematodes globally and are using that in a species distribution model in relation to things like climate and soil type to try and predict where the risk areas really are across our sampling space. So the data are going to tell us where the nematodes are, where they're going, what types of crops they might affect, and then that allows us firstly to focus in our sampling efforts to really find out how far they've gone to date, and then also to highlight the real risk areas across Europe where farmers and growers and national plant protection organisations can take action to prevent further spread.
We are centered in looking for a resistant gene against a kind of root knot nematode that is meloidogyne that could be stable at high temperatures. So what we have done mainly is compare with some genes that are not now in the varieties that are commercial, that is heat stable, that comes from a wild variety, and compare with MI1.2 gene that is present in the commercial varieties. With this, we found some parts of the genes that might be important for keeping this heat stability. And with this, we are going to construct in the next months markers to select heat-stable MI genes in different varieties. The idea is look for natural varieties that could be stable at high temperatures, which is very important in most of the countries, but particularly in Mediterranean countries where the temperatures of the soil increase to higher temperatures of 28 degrees, because the resistance of the gene that is now commercial and very effectively used is broken at this temperature. So it does stop working and you have infection in your crops when the temperatures are higher. And then on the other side, we are also working on identifying cofactors that could contribute to the heat stability or instability that are not only the direct resistant genes. And we have started to collect material to do epigenetics and expression profiles to try to identify these other candidate genes that could also help to make plants more resistant to the nematodes under high temperature conditions.
So far in work package 3, which is mainly interested in understanding how nematodes overcome plant resistance, we have obtained some preliminary results by comparing the genomes of some virulent populations that were able to overcome the resistance, and we found several convergent genomic variations. One of the most promising one is a big deletion in the genome of more than 1 megabase that covers almost 300 genes, and this gene seems to have been completely lost in the virulent populations. So we don't know yet if it's causative, it's just a correlation. Now we see these convergent deletions, so the next step is to confirm if these deletions are also present in field population, because the population we have studied so far are lab populations that were multiplied in the lab by experimental evolution. So we hope that the variation we found in the lab population will be confirmed in the field population. So far it's for just one species, Melidogaene incognita, but in this work package we have 3 other Melidogaene species, and for these ones at the moment we don't know. So the future will tell us, once we get the new sequences, if we find similar variation in the genome.
I'm leading the work package number 4. We have 3 tasks and 3 objectives. One about the nematodes, one about the plants, and one about the mathematical modeling. About the nematode, the objective is to identify the genomic region to the virulence of Globodera pallida. So during this year we have selected, collect different populations from the different countries, from the consortium of NEM-EMERGE project. We are now phenotyping those populations to quantify the level of virulence, and we will then sequence all the genome of those populations with a pulse-seq strategy, and we will, with this approach, identify genomic region and genomic SNP variant to compare the virulence. And the next step will be to develop a molecular tool to be able to follow the frequency of the virulence in natural population of Globodera pallida. In the modeling task, we have developed this year a first model to enable us to model the dynamic of the virulent adaptation. And this model will be now integrated into decision support system, into existing decision support system, such as the NEMA-Decide, or the one developed by the James Hutton Institute. And for the last task, for the plant size, we have decided to focus on the resistance from Solanum parsipilum, which is conferred by 2 QTL of resistance, one on the chromosome 5 and one on the chromosome 11. And we are performing a strategy with an enrichment in resistant genes to sequence different bulk with 0, 1, the other, or both QTL to identify a marker linked to those QTL of resistance. And those markers will be available for the potato breeders at the end of the NEM-EMERGE project.